see through a different lenses. We talked at length about what does this image show us in terms of the evolution of plants and their pigments. Because if you're starting to get into your project, you're going to see that these pigments kind of overlay into your plant metabolism. Because different metabolisms require different amounts of heat, different amounts of water vapor, so on and so forth. So don't get lost in the pigments, but also don't get lost within the statements of C3, C4, and CAM analysis. So let's go ahead and jump right in and see how you did on these statements. Here we go. Number 1, uses PEP carboxylase for fixation. You should have had C4. All right, C4. And that's really going to be one of the big statements, is that when we start looking at this, now I know that some of you are going to say, well, you said PEP carboxylase here, but really the mentality of this is that this is crustaceous acid, this is PEP carboxylase, and this is Rubisco. That's kind of the way you need to look at it, because this happens at night, it's stored. Then yes, PEP carboxylase does play a role in it, but that's not the primary fixator. It's going to be that crustaceous acid, all right? So number 1 should have been C4. Number 2, carbon dioxide stored as crustaceous acid, that is going to be CAM. Number 3, utilizes bundle sheath cells for the Calvin cycle. What'd you pick for this one? That's C4, all right, C4. Number 4, susceptible to photorespiration. That should have been C3. And that's kind of what we talked about yesterday, and I don't want this to get lost, is I mentioned it a couple of times, but it kind of maybe went in one ear and out the other. It didn't really land to some of you. Whenever we talk about evolutionary mechanisms and we look at anytime we do a comparison, we should always be asking, why are these things different? Whether it's photosynthesis for cell respiration or 3 separate entities within photosynthesis. And what we talked at length about yesterday were the trade-offs. We looked at the C3 plants, low machinery, not a lot of enzymes, not a lot of stuff going on. So it makes it pretty efficient, right? But the trade-off of that is what? The ability for the plants to undergo photorespiration. So an adaptive feature for C4 and CAM is to adapt compartmentalization, more enzymes, more machinery. So what's the trade-off though? So they're spending more energy to build all these compartments and things, but what's the trade-off? What do we know that these two do not do that this can? Photorespiration. So in evolutionary biology, there's always a trade-off. If you're going to build something and spend energy, whatever you get out of it, better and does overcome and have a greater impact than what you spent. This is that cost-benefit analysis we talked about with enzymes. Plants are no different in this endeavor. So the trade-offs, keep reminding yourself of the evolutionary implications of these. Okay, so number 4 was C3. Number 5, the first stable product is a C3 compound, or a 3-carbon compound. That is C3. That's literally what the 3 is for, a 3-carbon compound. Then you go to the next one. The first stable product is a 4-carbon sugar, oxaloacetate. Well, 4 carbons, that's going to be C4. That's where the names come from, is their first stable compound. So whether it's PGA for that C3 compound or oxaloacetate for the C4. And then, of course, you have crustaceous acid, which is going to be for your CAM. Okay, now let's go to number 7. Number 7, you should have had CAM. The context clue is that the stomatas are open at night. All right, then it goes down. Loses the least amount of water. I'm interested to hear what y'all picked for this one. Which one loses the least amount of water? See, I'm glad you said that, and that's okay. A lot of people want to say CAM, and some of you are probably like, well, I researched it. But let me debate this a little bit, and this is why. Just because these CAM plants open at night, that means they stay open longer. But don't forget, just because it's at night, aren't those places still dry? Right? The temperature might not be as high, but they're still dry. So water will still leave. So what determines the rate at which water exits is how long the stomata is open. So in my opinion, and in a lot of scientists' opinion, is that the plant that has the least susceptible loss of water is your C4, because of the time that the stomata is open. Because the logic wants you to think, well, it opens at night and it's not as hot, so I'm not going to lose as much. But the environments are still dry. Okay, so that's really, really important that we see that. Number 9, stomata open during the daytime. So you should add C3 and C4 for this one. All right, and then number 10, rubisco is part of the metabolic pathway. What'd you pick here? All of them, yeah. All right, and that's really why I threw that one in there, is because you notice once we get here, the Calvin cycle is the same. The only thing that's changed is the fixation component and how long those stomata are open and the compartmentalization to capture that carbon dioxide in a more expedient way and how it's stored. That's really the biggest difference. Once we get to the Calvin cycle, it's the same MO for all 3 types of plants. So very good on that one. Okay, number 11, the most common plant. C3, there we go. Number 12, requires large amounts of carbon dioxide, may benefit from global warming. I hear a vote for Cam. What else? C4. Anybody pick C3, just to round this out? All right. So I've got, all right, we got to vote here because I'm interested. This is, because whenever we talk about climate change and global warming, I'm always curious to hear what people think. All right, because of where you're getting your information from is sometimes questionable. So how many of you picked Cam for that question? That is best suited for global warming or climate change. How many of you picked C4? Okay, there's a good handful of you that are not voting. All right, so let's try that again. Cam, you have to pick. Put them up high. Let's go. All right. Okay. C4. Wow. Almost half and half. All right. You said Cam first. Tell me why. Give me some logic.
Cam, the plants that have cam photosynthesis are in like really extreme types of temperatures.
Okay.
So therefore, if that outside temperature was to get warmer, then cam would probably be the one that would adapt and probably survive the most.
Okay. All right. Good logic. So who said C4? Who said? Yeah, you're hiding. Look at her. She's like, no, don't call on me. Why'd you say C4? If you don't mind me asking.
Well, because the C4, like, they need lots of carbon dioxide for the carboxylase to bind to it.
Mm-hmm.
So that's why I said it because it said it requires large amounts of carbon dioxide.
Okay.
Oh, wait, we talked about this yesterday.
Mm-hmm.
It was because the reason why global warming happens is because carbon dioxide is a greenhouse gas, so then its presence in the atmosphere already increases the temperature, and so therefore the cam plants would already have enough of carbon dioxide to fixate, so they would benefit more.
Very good. And to be honest with you, there really is no good answer. There is. I'm not going to stand in front of you and say this one over this one. But I put it out there because I wanted to see what your logic would be. Because if climate change is real, and it is, because climate's been changing for billions of years and will continue to change, but at what rate and things like that, we do have to speculate what could happen. We all agree that C3 plants are not going to, they're the ones that will not adjust very well. Whereas if I were to make the argument, I think both of you make good points. Let's argue the C4 for a second. I think C4s would be very, very well adapted because of the time that they only keep their stomata open. And they have the ability to fluctuate between drastic temperatures. Because here's the trick, and this is what you have to remember. Global warming and climate change actually triggers what? Global freezing. So eventually, you're going to get into colder temperatures. So does that benefit the cam plant or does that benefit the C4 plant?
Cam.
Potentially, because cam has the ability, did anybody read about cam idling? Did anybody come across that? Where the cam plants have the ability to shut themselves down just for kind of like a polar bear hibernating. Cam plants can do that. So I know we're going down the rabbit's hole here, but it's kind of interesting to talk about. But I'm not going to get up here and profess in front of you to say it's this one or this one, because I honestly don't know. But we use the logic and we can speculate from there. So good conversations there. Let's go to number 13. You should have picked C4, all right? And some of you are saying, well, wait a second. Isn't malate and cam as well? Yes, you should have also picked cam in there as well. So when you have that malate, that's going to give you that secondary reactions. let's go to number 14. Stomata open for less time. Carbon dioxide brought in much faster. That was C4. Number 15, the most efficient. What'd you pick here? This is tricky. C3. Most efficient is C3 because it has less enzymes and less machinery. Process occurs throughout the entire leaf. C3. And that's kind of when we were talking about the compartmentalization. C4 and CAM plants have compartmentalization. As you can see, I've added some notes here for you. So this compartmentalization, that's the trade-off. That's what I keep mentioning to you. By adding these, the trade-off is, hey, I'm going to spend a little bit more of my ATP to build this. But what I'm going to get out of it is I'm going to reduce almost to zero the chance of photorespiration. So it's worth taking that chance. All right, so that, because you got to remember too, these are also in extreme environments. So they're going to want to keep those stomata closed, right? Kind of what causes photorespiration here. So that's why they have that extra compartment so the stomata doesn't have to stay open nearly as long. Okay, number 17, maize, sugarcane, sorghum. You should add C4. All right, a lot of your commercial crops, but then we go into cash crops. Like if we go down to 19, cotton, peanuts, rice, tobacco, pretty much the cash crops of the South. That's all C3. But if I go back up to 18, pineapples, aloe vera, cacti, those were CAM plants. Okay, and then number 21, believed to be the first to evolve in the plant kingdom. We talked about this yesterday. That was C3. Seasonal plants found in cool, wet areas, C3. Habitat, tropical environments, those are C4s. And then your deserts are going to be your CAM. Okay? Any questions, or those need to be repeated? All right, very good. Now, like I said, most of these, when you're doing your project, which, by the way, for you is due Thursday, all right, when you turn in your project, these are the types of things I expect to see. Some depth, some detail. The rubrics are posted for you on your Canvas page, so I'm expecting to see some thorough thought. And then, of course, don't forget, as I mentioned yesterday, adding in the pigments. Talk about the evolutionary implications. I'm looking forward to reading your kind of opinions and your ideas about how these plants have evolved. And more importantly, when we start getting into ecology, I do a project where you are assigned an ecosystem and we fast forward 20 million years from now. And you have to speculate of where that continent's going to be and the plant and animal life and how has it changed, if at all. Okay, so a lot of things that go through there. All right, so this is where we're going to go through today, and we are going to go back to the basics. Photosynthesis 101 starts today. So go ahead and get out this image for me. All right. And second period wanted me to use purple, but I didn't think it was showing up very well. So I'm going to go with, I think I'm just going to stick with black. That's probably going to show the best. Let's see. You guys can see that, right? Okay. Let's go with black. All right. So here's the thing. I'm going to make an assumption that you know nothing. Don't take that as an indictment, because I'm sure a lot of you remember this. But sometimes when people come from different classrooms, different teachers say things differently, they use different vernacular, they think one thing's more important than the other. I'm just going to throw it all at you. All right, and just make sure you understand the basics of this. So one thing I always tell students is always work backwards. Think about each process and why we're doing this in the first place. Why are plants going through the light-dependent reaction first? The whole premise is to build ATP and NADPH. So these are what we call, all right, let's try that again. Okay, these are what we call electron carriers. Okay, these are both electron carriers. So that's the premise. And maybe this is just the way my brain works. I like to work backwards. Say, okay, I know where I'm headed. I know what I need to do. But how did we get here? So we're building ATP and NADPH. Those are electron carriers. So where are those electrons coming from and what are they doing? So the first thing I tell students to always look at is look at where we're at. We are looking at a thylakoid inside of a chloroplast. Now, stacks of thylakoids, if you learned your freshman year, we call those grana. All right, that's just kind of a fun fact to know. Nobody's really going to ask you about that. And then the quote-unquote cytoplasm of the chloroplast is called the stroma. So see right here where it says chloroplasm, write stroma for me, just so we're all clear.
Says stroma at the bottom.
Yeah, I don't know why it says chloroplast. I don't know why it does that, but just so there's no confusion. I always use stroma, just to be clear. No, I've never heard any professors. I've never even really seen the word chloroplasm. So I don't know who came up with that. It's like science, just mashing words together. So one thing I want you to pay attention to is the pH difference between the stroma and the thylakoid space. Because we talked at length about how delicate pHs are and the measurement of hydrogen ions. And that's what pH is. So when we look at the stroma versus the thylakoid space, we can automatically identify that the thylakoid space is going to be much more acidic. So therefore, it's going to be a higher level of hydrogen ions inside the thylakoid space versus the stroma. Now, you're probably wondering, why do we care about that? Why does that matter? Because this right now already establishes our proton gradient. That's a very, very important thing, is that proton gradient is going to be determining what's about to happen. So we've established that the thylakoid space is highly acidic, lots of hydrogens, so we have our proton gradient. Now we get into where the electrons come from. And what we said yesterday is that's the purpose of water. Water is our electron donor. So water is going to go through hydrolysis, it's going to break down, it's going to release oxygen gas, as you can see right here. Now, one thing I'm going to tell you that I want you to pay attention to is oxygen gas just diffuses out immediately. So when we look at our photosynthetic formula, we see water to oxygen gas, but as we've already identified, oxygen gas is a waste product. So that's got to be eliminated right away. So that's off the board. We don't even worry about that right now. But we do have hydrogens that are remnants of that. But really what we care about are those electrons. Those electrons are going to enter in what we call photosystem 2. Now, some people often wonder, why is the electron transport chain in this order? Why is photosystem 2 before photosystem 1? It's all based on how it was discovered. Photosystem 1 was discovered first. Then they kind of started looking and said, something's not adding up, and they started working backwards, and then they ended up finding photosystem 2. Photosystem 1 has what we call P680 chlorophyll molecules. Now within this thylakoid, those chlorophyll molecules, that's going to have your chlorophyll A's, your chlorophyll B's, all right, your carotenoids. And those are going to be what's absorbing all of your radiant energy that you can see here. Now, the reason why this P680 is first and then your photosystem 1 is P700 is your P680 is much, much more powerful because it's attracting the water. It's attracting those electrons. And this is why we've said in the very, very beginning, what type of environment do plants always want to exist in? Isotonic, hypertonic, or hypotonic? Hypotonic. Because without water, there's no electrons. With no electrons, this doesn't happen. You can have all the light in the world, but without water, there's no electrons. So this is why for civilizations have sacrificed live animals, sometimes people, all right, and do weird dances around campfires and pray to whoever they pray to for rain because of this. Now, they probably didn't know you need electrons, nothing like that, but they knew they needed water. Now you know why. You need those electrons. So here's what happens, real quick and simple. The energy of those electrons is going to increase because the light that is captured by those P680 molecules, it's going to energize those electrons. So the energy of those electrons is going to rise. Now, why? Why do that? Well, those electrons are then going to be relayed through a series of electron transport proteins. Now, you don't need to memorize these, but I don't know, you know me. I like to throw out some extra credit questions once in a while. This protein right here is called plastoquinone. We know cytochrome C. This is plastocyanin. Now again, you don't need to memorize those, but it's just always fun to throw some big words around and sound really smart, right? So really what's happening is that this is just the electron transport chain. Why transport electrons? Why does it do this? Well, here's the big takeaway. Between photosystem 2 to photosystem 1, a process occurs known as chemiosmosis. That's terrible handwriting. All right, chemiosmosis. Now, chemiosmosis is what's going to be driving the production of ATP. Okay? Now, what this means is that once those electrons become highly energized, they become highly energized simply to lose that energy. So from photosystem 2 to photosystem 1, we call it the fall. So as electrons are passed, every time they are passed from protein to protein to protein, they lose energy. So by the time they get to photosystem 1, the energy level of those electrons is extremely low. But if I've taught you anything, you should be asking a series of questions. Where did the energy go? Right? So we have to be able to answer that question. So the energy was the electrons are highly energized here, and then by the time they get there, they lose that energy. Where did it go? You already know the answer. Follow the hydrogens. The hydrogens are going to be pumped into the thylakoid space. Is that active or passive transport? What do you think? It's active. Tell me why. Well, okay, yeah, I told you if it's pumped, it kind of, that's good. But what was your other evidence? It does. The pH, exactly. So remember we said this was low hydrogen ions, these are high hydrogen. So we're literally pumping those hydrogens from the stroma into the thylakoid space against the concentration gradient. That is active transport. So chemiosmosis, simply put, is evidence of active transport. So the sunlight in photosystem 2 that's captured energizes those electrons. As they're transferred, that energy is lost for the sole purpose of pumping hydrogen ions into the thylakoid space. Now here's the fun part. Now we get to see the reemergence of an old friend. Follow your hydrogens. Where do they end up? There it is, ATP synthase. All right, putting you on the spot. How many hydrogens do you need for one ATP? 3, very good. So 3 of those turn, you get 1 ATP. That's kind of way back. That was a long time ago. What was that, August when we did that? Fun times. All right, so now you get ATP production. So we are phosphorylating ADP into ATP. So this entire process, yes, from photosystem 2 to photosystem 1, we call that the movement of hydrogen ions, chemiosmosis. This whole thing, all right, you ready for this? This is called linear, all right, I'll write these out for you. This is known as linear photophosphorylation. Linear photophosphorylation. Linear means straight, photo means light, and then phosphorylation means the addition of phosphates to energize something. So because of chemiosmosis, hydrogens move in, and then they move through ATP synthase. There's your first electron carrier, ATP. But we're not done yet. Now, more sunlight is captured by the P700. Not nearly as strong, but enough, right? And so all of a sudden now you get these electrons, and they're going to move to this protein, FD, known as ferrodoxin. This is ferrodoxin. And then the last thing, and I tell students this all the time, a rule of thumb that you always have to follow. If there's an electron donor, you always have to have a final electron acceptor, no matter what. Because you can't just have electrons floating around willy-nilly. So if the water is the electron donor, what's the final electron acceptor? And that is what you're looking at right here, known as NADP plus reductase. So that electron kind of terminates there. And with that energy, because remember it's highly energized, it's going to phosphorylate into NADPH. Now, like we've already identified, those are electron carriers. And you notice all of these are being built on the external surface of the thylakoid membrane. So now both of these are in the stroma. Now, does anybody want to take a stab and tell me what NADPH stands for? We know what ATP stands for, hopefully. adenosine triphosphate. That's an easy one. You're trying to tell me they didn't teach you what NADPH stands for? I'm kidding. They had no business doing that. All right, you ready for it? This is an extra credit question. You never know. Okay, NADPH. Here we go. It stands for nicotinamide. Nicotinamide. No, we're not. That's only like a quarter of the way through. Nicotinamide, adenine, dinucleotide, phosphate. Say that 5 times fast, huh? Nicotinamide, adenine, dinucleotide, phosphate. That's just a fancy way of saying electron carrier. Now, let me give you a rule of thumb, too. Little helpful piece of advice. When we get into cell respiration, you're going to see NADH, which is just nicotinamide, adenine, dinucleotide. That's it. There's no phosphate there. So this phosphate, the NADPH, you only see in photosynthesis. So just remember, if you see the P there, that stands for photosynthesis, even though it doesn't. But that's just a context clue to help you remember. NADPH is only in photosynthesis. So now we have these 2 electron carriers. Here's the good news. We're halfway done. Here's the bad news. We're only halfway through. All right, so we now need to get into the Calvin cycle. But before I move on, is there anything about this that you have questions on? Hit me.
So if NADPH stands for whatever that was,
You don't want to say it?
now, what does NADP plus stand for? Is it just phosphorus
Mhm.
plus? Okay.
Yeah. Okay, any others? Now, like I said, you don't need to memorize all of the proteins. Now, listen, if you just start throwing around those words, you sound really smart and really, really cool. I don't care what anybody says. All right? But the fact of the matter is that the whole purpose is when you see these, think. We talked about the other day, preservation of water. The capturing of light in the evolution of the different pigments. So all of these plants have evolved different ways of moderating their water, capturing their light, and then, of course, running through the light-dependent reaction. All right? Any questions? Are we good? We sure? All right, let's move into the next one. So now what I need you to do is, and this will be much quicker, is go to this image here. Now, I don't care, because I've taught honors bio at the freshman level for years. And I don't care who you are. When you saw this as a freshman, that was scary sauce, isn't it? You know what I mean? That was intimidating, as all get out. Is this the picture they showed you, or is it a much easier one? Is this one? Oh, good. All right. You didn't see this one? All right. Yeah, for some people, this is like nightmare fuel. But the reality of it is, it's not as intimidating as it looks, because some of these things I've already taught you. So this is where we start getting the challenges, adding all of these things that we already know, whether it's pH, variables, phosphorylation, even the names of things I've taught you. So the very first thing that we're looking at is the Calvin cycle. And as we've already identified, regardless of how the carbon dioxide is fixated, all 3 types of plant metabolism end up here. We're in the stroma, and I need to change this because it says dark reactions. I don't like that. I don't want people to think that these only occur at night. These occur all day, all night. This thing is constantly churning. Assuming that is carbon dioxide is present, all right, and your electron carriers are present. But in this case, you have your carbon dioxides, they enter into the Calvin cycle. This is a cycle. So this is called cyclic phosphorylation. All right, your light-dependent reaction was linear photophosphorylation. Now, because light has no bearing on this, this is called cyclic phosphorylation. We're still phosphorylating, as you're going to see here in a second. But let's work backwards. What are we trying to build? And if you say glucose, I'm going to throw something at you. That's not what we're building. We're building a 3-carbon sugar known as PGAL, phosphoglyceraldehyde. Now, we know that if we take 2 PGALs and put them together, we can get a 6-carbon sugar, right? We know that. But what if I take more PGALs and stitch them together to form a large polysaccharide known as cellulose? So this isn't just for energy purposes, it's also for construction, for the plant cell wall. All right, let's dive in. So we know that Rubisco is that enzyme that fixates that carbon dioxide. All right, and all of these enter into this, so Rubisco is present in all 3 as we've already identified. What you don't need to memorize, but you do need to understand the methodology, is how do we get from here, to that 3-carbon sugar? Well, 3 carbons, all right, 3 carbon dioxides come together to give you a 3-carbon compound. As you can see, it's called 3-phosphoglycerate. Why the 3? Remember the naming? Remember when I taught you about names? Remember 2-deoxyribonucleic acid? What's that?
3 carbons.
Well, yes, the 3 doesn't tell us the 3 carbons, though. What does the 3 tell us? Exactly. So look at the number. So if this was if this was carbon 1, 2, and 3. So yes, you are right, Essen, there are 3 carbons. But this 3 tells us on the 3rd carbon, you have a phosphate. That's what the name, anytime, remember I told you, anytime there's a number in front of a chemical name, it's giving you a GPS coordinate of which carbon you need to look at. And how do I know this? Well, look, 3-phosphoglycerate, you don't need to memorize that, don't panic. But just look at the structure, there are 6 of these. Then all of a sudden, there are my ATPs. I'm not going to keep your feet to the fire of how many ATPs, because you need 18 ATPs here and 12 there, and you make 6 here. It just gets messy, and it's kind of irrelevant to what we're talking about, because this thing is constantly turning. So the 6 ATPs, they phosphorylate what? They're going to phosphorylate this 3-phosphoglycerate and turn it into 1,3-bisphosphoglycerate. So the only thing that's changed is what? We've added a phosphate to the 1st carbon. So by phosphorylating it, that's why I call this cyclic phosphorylation. I'm adding a phosphate to the 1st carbon. There was already a phosphate to the 3rd carbon. And you already know this. Anytime I add phosphates, am I increasing or decreasing energy? I'm increasing it, right? And that should be the goal of this, because we've already said this entire process, all of it, is endergonic. All right, it's going to be anabolic. We're building something that is high energy, low entropy. So photosynthesis is lowering entropy within its system because it's organizing all of that sunlight and building an energy-carrying molecule. So anytime we phosphorylate, we're adding energy. So not only does ATP do that, but then NADPH does the same thing. All right, it's going to add more of those electrons and keep building and building and building to eventually we get into this section called some teachers call it G3P. I don't. I call it PGAL, phosphoglyceraldehyde. So everything that you just saw, and what I want, don't miss this, right here. If you need to underline it, highlight it, point to it, I don't care. But we call that the reduction phase. Does that make sense to you now, what reduction means? What is reduction? We are doing what with electrons? Come on. We're gaining electrons. So now this phase should make sense to us, because all those electrons that we started all the way back with water, those electrons are still here. We're just transferring them around. So now we're taking all those electrons that were carried by ATP and NADPH that we built in the LDR, and we're using them as coupling reactions. Now remember, a coupling reaction says the product of one reaction, the products become the reactants in the next one. So in LDR, we built ATP and NADPH. Now in Calvin's cycle, those are reactants. So we carried those electrons from LDR in here, and we're just adding, like putting Legos together, adding them in to build or reduce this P-gal sugar. But because it's cyclic, what do we have to do though? We have to rebuild what? We have to rebuild Rubisco. So that's why we make more ATP than we do NADPH. There's always more ATP than there is NADPH because you need the ATPs to rebuild Rubisco because it is cyclic phosphorylation, and this process keeps turning and turning and turning. All right? So this is why I've shared with you that ATP isn't always just the energy that gives you energy. It's, in this case, it's an electron carrier. It's going to donate those electrons, all right, in this process. And then as a result, you get your 3-carbon sugar. All right? Any questions I can answer for you here? All right. So That is photosynthesis 101. Now, how much of that was review and how much of it was brand new to you? How much of it was brand new? A lot? Kind of, sort of? All right, how many of you have heard a lot of this before? Oh, good. Okay, so it is a good amount of review. So like I said, if you've heard it before, it just reinforces it. If you have no idea what I'm talking about, you're getting on the same level. All right, so we're all good now. Now this is what I want you to do. I'm going to back away because I know you're tired of listening to me, and to be honest, I'm tired of talking. What I'd like you to do is go ahead and look at your packets, and you are going to accomplish, however you want to do this, several different things. The first one is you're going to work on your practice problems. However you go about managing that, your big boys, big girls in here, I'm not holding you by the hand anymore. You know what you need and how to get there. So if you want to do these without looking at the answers and then go back and look at the answers, awesome. If you want to look at the answers first and then kind of examine why that's the correct answer by looking at the questions, cool, totally up to you. Those answers are posted on your Canvas page. Then I also want you to complete the graphic organizers, the photosynthesis sections of those graphic organizers. And if, like I said, if you're feeling squirrely and spunky and you want to work on the cell respiration part, do it. But the last thing, probably not overly necessary, like priority-wise, because if I know you the way I think I do, most of you will probably do this tomorrow night at some point. The pre-lab with our lab is going to be a little different. When you go to your Canvas page, you will see, I've written a note there, there are what we call focus questions for Investigation 1 and Investigation 2. All right? So you're going to do the focus questions, but you're not turning that in on Thursday. Everybody got that? You're just going to save it and turn it all in with your final lab report. However, it is absolutely critical, absolutely imperative that you read and you complete the focus questions before you come to lab on Thursday, because if you don't, you're going to be the chump that has no idea what's going on. And nobody likes that lab partner. And look at y'all looking at each other. You guys always call out the person without even saying it. You look at it like, yeah, that guy. All right, don't be that person. Okay, make sure you're reading. It doesn't take long. It's not a lot of reading, but just understanding. We're using microscopes. So we're doing two different investigations. One of them is microscopy.